You got it?
Okay, started.
Sure. Okay, so thanks everybody for coming today. We're really excited to have Marianne Bauer. Marianne is a new assistant professor at Delft University of Technology in the Netherlands in the Department of Bio-Nanosciences. She's focused on improving understanding of behavior at different spatial and temporal scales by sort of analyzing simple physical and statistical measures or models. She most recently was a postdoc at Princeton as a fellow at the Center for the Physics of Biological Function, working with Eric Wieshaus and Bill Bialik to use kind of like information theory concepts to figure out how different input signals could yield precise spatiotemporal expression. I think that might be what we're going to learn a little bit about today, which is great. Prior to that, she collaborated with Erwin Frade, a model. She worked on like ecology problems, modeling the emergence and stability of biodiversity. That's probably, I think that was the first time you really did like biology stuff. Right. Seems like your PhD in physics was like mostly studying Fermi gases, really hardcore like theory stuff that I'm scared of.
Maybe me too, I don't know.
But we're very excited to hear from you today. And without further ado.
Yeah, no, thanks so much. That was a super kind introduction. Thanks very much for having me, Simon, Karna, and Manav, I guess, for the invitation. I really appreciate it. So I guess, as Simon said, I moved to biophysics a little late. And I guess I just started at TU Delft. So I'm going to talk to you about this like last project that I've done in Princeton. And I really enjoyed that you also tripped over the CPBF, the acronyms for the center that I was working on at Princeton. It took me probably 3 years to spell it correctly. So
they got to get a better name for that institute.
it's tough at the P and the B. I don't know.
Exactly.
But so thanks for the intro. And so I guess I'm going to talk to you, as someone sort of anticipated, I'm going to talk to you about this interest in sensing transcription factors in the fly embryo that's, I guess, started at Princeton and that I've, I guess, done in Princeton in collaboration. But I should say that it's sort of interesting to me because for me, what I enjoy about this, actually, let me check. I think maybe can I move forward? 1 second. I might have to go back out and screen share again because I think I can't advance the slides. I'll try to get this figured out.
Does it help if you click on the slides at all?
Yeah, that's what I'm trying, but I think oh yes, okay, perfect. I think, let's see if it works out. Yeah, okay, great, perfect, thank you. Okay, so it's just the mouse that isn't working. Okay, so what I'm interested in is this general question of like sort of, what makes biological systems precise and when are they reproducible, and sort of also what tricks do they use when we observe reproducible or precise behavior? What tricks do they use to stabilize this behavior? And so gene regulation is obviously one big question where behavior needs to be precise because the genes need to be regulated in ways that the organism can later on use to develop in healthy into a healthy and functional form. And so that's an aspect where you really want precise behavior. And so I'll talk to you first a little bit about gene regulation, this question of sensing transcription factors and how gene regulation works. And then I'll sort of introduce and give you the outline for the talk. So gene regulation in principle is this, broad question of how do genes get expressed in ways that make an organism functional. The traditional idea that in biophysics that we had of how this might work is sort of inspired very closely by the lac operon. So what you have there is like a binding site array where the transcription factors can arrive, they can bind. Transcription factors in this talk, I should say, are always, the concentration is always indicated by the small g. I know that's a bit confusing, but the idea is basically that in a transcription factor network, you start with a gene that's expressed, and then it basically becomes a transcription factor. So when I talk about the gap genes later, I always mean the transcription factor, so the protein that leads to the downstream expression of the gene. So this transcription factor can bind to a binding site array in the lac operon in bacteria that's like close to the promoter of the gene that's being expressed. So the promoter turns on, the gene is expressed, and it's a simple idea. But now we, of course, know or have known for like several decades now that in eukaryotes, the system is a lot more complex. So there, what I've tried to sketch here is that there are multiple enhancers. These enhancers can often regulate the same gene. They can regulate multiple genes. They are often several kilobase pairs away from the gene that they regulate on the DNA. So already here, the simple idea with the lac operon is difficult and doesn't really apply. And then also more recently, there's been this whole question of liquid-liquid phase separation. So what this is, without talking about mechanisms, what this in principle corresponds to is this problem that transcription factor concentrations are extremely non-homogeneously distributed. So you might have clusters, and that means that basically when you observe the transcription factor concentration at some point, somewhere averaged over the cell, it might still not give you a good idea for what the local concentration in the region around the binding site is like. And so that makes it very hard to say something about gene regulation or gene expression, compared to the lac operon, where the system is like a little bit more simpler. And so with that kind of broad interest in mind, how can, these complexities maybe help stabilize gene regulation? I started this project in Princeton, and so I should, I guess, start with the acknowledgements. So this project was always done with Bill Bialik on the theory side, and then Eric Wieschaus, who's provided a lot of input, a lot of biological questions, and then Mariella Petkova, Thomas Gregor, and I guess also Eric Wieschaus on the experimental side, who did the measurements for the data that I'll be using. This talk, so in the next 45 minutes, and I should say, interrupt me at any time for questions, but I'll also break, I think, at like around 3 or so times throughout the talk, so that maybe, then questions that have been built up can be asked, but also please feel free to interrupt me. So in the next 45 or so minutes, I'll talk about this approach that we've tried to use on the fly embryo that's different from, I guess, the traditional lac operon modeling. So I'll first, and this approach is an information theoretical or an optimal sensing-based approach. So I'll first introduce why we thought this approach was useful. So I'll talk a little bit about other approaches, and then in this one in particular. Then I'll introduce the biological system, so the data for the fly embryo. I'll introduce this picture. And then I'll sort of jump ahead. I guess I'm jumping ahead now, but in the end, I'll talk to you about what we find. So essentially, what we find is that an optimal sensor that senses these transcription factor profiles needs to be structured in a way that actually practically would correspond to multiple sensors. And also practically what would be important is that these sensors should be split efficiently. And I think that's cool because basically the idea when you think of fly transcription, even within the first hours, is that because of these enhancers, the situation is really complex. But what's nice here is that we applied an extremely abstract approach. And we found that if you think of these sensors as individual enhancers, we found that actually multiple enhancers would be necessary, and that these enhancers need to have a certain structure. We haven't even put in any of the specific details of the biology. All we had was the transcription factor profiles. So what I think is nice is that you can actually start with something that's quite abstract and still learn something about the biology, still on a fairly conceptual level, but it's still nice. And I hope I'll convince you that is true and that is exciting. And so with that, I'll start with the background. So what I'll have the first couple of slides on is these sort of traditional standard models for gene regulation inspired by the lac operon. So what was important there, what people typically thought about when they thought about precision and in gene regulation, is that you have this input concentration G, and you have an output of a product. And now the question is, what happens if there's a sensor like this binding site array in between, which mechanistically needs to be in between? What kind of models do we build for that? And the first class of models, I would say, is broadly in this group of what I would call occupation models, which is a vague term, but the idea here is that the output of the downstream gene is proportional to the occupation of the binding site. This binding site region I'll from now on always refer with this letter C. And this entire group of occupation models, I would say, is a group of models that have been pushed forward by people around Rob Phillips, people around Ulrike Gaul, and then recently, sort of from a theory perspective, Jeremy Garner-Wardena at Harvard and maybe Angela DePace from the experimental side. So, I mean, obviously they've done very, great and also much more detailed work than what I'm describing here. But I would say that the general concept here is this idea that you have a region of a few binding sites, and that the occupation at this binding sites increases in some way or another with the input transcription factor concentration G. So you see that here, C is on the y-axis, G on the x-axis. You have some mean and some noise around that. the nice thing, and this is something that Jeremy Gunnar-Wardena has also shown recently, the nice thing here is that this Hills function, so the function that I'm showing here, that is basically the steepest possible way you can have the occupation at this binding site region increase when you have H binding sites. So what you need to imagine is that you have H binding sites, and now assume that you can't have one transcription factor bind and then the next one, and so progressively the binding site measures, but like instead all of these transcription factors bind at the same time. That's the steepest possible activation curve, and that's important because I think as a theorist it's always nice to have limits. So it's nice to know that that is the steepest one, so you have some sort of bound. Another group of models that I want to come back to also frequently in this talk is sort of originally, or has been discussed in the fly embryo literature, sort of originally in the 1970s, and that's what I would call, or like, I guess, an abstract fly gene regulation model that's based on thresholds. So many of you might have seen this picture. This is the typical French flag picture, and the idea here is that the gene is basically expressed when the transcription factor concentration is higher than a certain level, then the downstream gene is turned on. When the transcription factor concentration is lower than the level, then the gene is turned off. Now, if you have a gradient of transcription factor concentration, then basically you get sort of a stripy pattern if you have multiple genes. The idea, like this was very broadly discussed, especially in fly regulation or morphogen gradient regulation. But the thing that's always a little bit irksome about this is that in principle thresholds are associated with zero noise. And so that's, of course, when you think about the biological mechanism, it's a little bit difficult to think like that. So the question is, how can we make that more realistic? And so people said, well, if we know that the Hill function is the steepest way of activation in this occupation model, we can think of the Hill function and we can think of like the noise in binding then, and then we can basically say that a very steep Hill function corresponds to a threshold. So let's think about the noise in binding just really quickly. The noise in binding, and I think it's kind of interesting that the noise in transcription factor binding is still sort of inspired by a bound that was developed in the 1970s for chemotaxis, where the question was that bacteria had to sense again a gradient. So now this is applied to transcription where we need to sense a concentration gradient. And this Burke-Purcell bound was refined in the early 2000s, again, by my advisors, so Bill Bialik, Ned Green, and then also Peter Rentenwolde and Alvov and many, many others. And the idea here is like, how precisely can a particular binding site region measure this concentration gradient if you measure for a particular time, here denoted by tau, if the measurement device that the molecules bind to the binding site has a certain area A, if the molecules diffuse with a particular diffusion constant T, and so on. And I don't want to go into the details of this expression, but what's important is that they found that when you consider more and more mechanism, the Burke-Purcell bound is still the lowest possible bound for noise for these arrival statistics of these molecules. But so what you can see now is that this noise bound depends on G. So you need to have a local measurement for the transcription factor concentration in order to estimate this bounds correctly. And then there are also these other parameters that maybe are easier to measure, but it basically depends on a lot of parameters. And so that's essentially the problem here, that these models are difficult to use because the noise is kind of unclear and it's extremely hard to measure in experiments. I'm sorry, this happens all the time. I'm gonna mute myself.
Dames en heren, dit pand sluit op werkdagen om 8 uur 's avonds. Na dit tijdstip is het pand alleen
This is
nog
basically our
voor
wake-up call
studenten
to leave the building because it's Europe. I'm
Wij
sorry.
willen u vragen om het pand te verlaten
I'm just
als u
gonna,
deze toegang niet heeft. Dank u wel.
and now it's gonna
Ladies and
repeat in English.
gentlemen,
Just
this
maybe mute me
building
for 1
closes
minute.
on
I'm sorry.
weekdays at 8 p.m. After this time, the building is only accessible to staff and students with 24/7 access.
Sorry.
We would like to ask you
Like,
to
they
leave the
don't
building
really work so
if you
hard.
do not have this access. Thank you.
It's intense. It's intense, but it's like European working regulations. So, I shouldn't complain. Okay, sorry about this. Anyway, so I guess where I got to was basically this point that we can estimate the noise using the Burke-Purcell bound in something like the occupation model, but we can't measure the parameters, and so that's why these models are so it's extremely difficult to apply these models to gene regulation. But the approach that we tried to do at Princeton was to basically use an idea from information processing and neuroscience to these systems, apply these approaches, and then basically see what we can learn. And so in the next couple of slides, I'll introduce this other approach, hopefully without another interruption. So this inference approach that we've been trying to think about is basically one approach where you try to infer the optimal sensor. So you don't write down a model for it, but you initially have no model at all. You just have essentially a black box. You have a sensor that you describe by this variable PC, and then you say, well, the sensor is fully described by a probability distribution PC given G. So PC given G now describes your sensor. That's the thing that you try and infer. And then the nice thing is that you can compare this PC given G with the, I guess, what the equivalent probability distributions would be from the models. So you can compare it, and then you can basically, based on that, sort of say which model would most likely describe this optimal sensor. So, you know, the threshold-like model, I guess, would correspond to a PC given G that only has a few separate Cs. So in terms of the variable space, it's a reduced cardinality and these sharp thresholds, whereas, of course, the occupation model has this mean and the noise around it. So that's the direction we're going. But then, of course, what you need is G. And so on the next slide, I'll say a little bit about how we kind of can infer this optimal sensor. So in order to find an optimal sensor, we need data. And the other thing that we need is some intuition of what component of that data is important. So what component of the data does the embryo actually need in order to develop healthily, or, you know, for the gene expression to be precise. So this variable of importance, you know, I'll explain later on when I introduce the data for my system, but in general, we can call it X. And so then what this optimal sensing scheme essentially corresponds to is a compression of the data so that the relevant variable remains extremely well readable, so that the compressed sensor, the sensor C, has a good intuition of what the relevant variable is, but that the data is compressed. And just to make this a little bit more concrete, because these methods are also used a lot in machine learning recently. So if you have data that's basically corresponding to an image, then what you don't need is all of the individual pixels, but what you might want to know is what is the image content. So that's sort of the relevant variable X. And so you want a compression of this image in terms of C that keeps the relevant data. There are a broad number of such compression schemes, so you can imagine multiple ones, of course, but they typically need a loss function or some optimization goal. And in this work, we've used an information theoretical optimization goal. I don't know, you know, what the background here is, or like, you know, but whether people have already seen the mutual information before, but I've written down the formula here. The I guess intuitive point is that the mutual information is 0 when two variables are not correlated at all. So assume you look at one variable. If you get 0 information about the other variable, that's when they're uncorrelated, when the mutual information is 0. And if you observe one variable and you know everything about the other one, that's when they are extremely well correlated and the mutual information is its maximum, which in this case corresponds to the entropy of one of the two variables. So I mean, it's a technicality, but for the rest of this talk, I'll come back a lot to these mutual informations, because the advantage is that as a theorist, many, you know, ways of writing down probability distributions can be done analytically when you use the mutual information. And so that's why we're using it here. And so the scheme that we use abstractly basically corresponds to this information bottleneck scheme. I'll explain it more on the data, but what it corresponds to is basically that we want to maximize the information between C and X. So that basically tells you that the sensor, the binding site array, should have a great, a great correlation or a very good idea about the variable that we care about, whatever the system needs for healthy development, for various different mutual informations between C and G. So what this means is that we want to essentially leave it up to the sensor, or because we don't know how noisy the biological system measures, we want to vary through the noise essentially and say, you know, for a particular mutual information between C and G, let's find the best optimal sensor and let's look what does this give us about X. And this information bottleneck scheme has recently been used a lot in machine learning, but was also sort of developed, of course, early on. But so the idea is that this goal basically corresponds to something that's a sensible optimization goal. And I'll explain why that is true for my system in a few slides. And so with that, I want to introduce the biological data. And actually, maybe this is where I should break for questions. So if there are any questions so far, I know it's been technical and it'll become more clear later, but maybe I should break here. Okay, then if there are no questions, that's great. Then I'll go on and
stupid one. What was the T on the previous slide?
Yeah, good point. So that's a Lagrangian multiplier, and the idea so the idea is that you basically don't yet know what noise is good for the sensor because it's abstract. So you don't necessarily know with what noise C should read G. And so with this Lagrangian multiplier, you tune through. So you basically allow for an entire range of different possible mutual informations. But I think it'll become more clear when I show the plots.
Interesting. Okay.
I also have a question.
Mm-hmm.
What is the, can you explain in a different way, what is the rationale between this information bottleneck formula?
The rationale behind the information. Okay, so basically the rationale is we have data and we have something that we care about, and we now want to find a variable that measures the data such that, you know, the information about the thing that we care about is as good as possible. So what we do is we write down an optimization goal, which says the sensor should optimize the mutual information between, you know, towards the thing that we care about. So optimize I, C, and X, while keeping fixed for various degrees of fixedness the information between C and G. So that basically corresponds to that the sensor can measure noisily, or it can measure less noisily, and so on. And so for various degrees, we want to optimize, so we keep that fixed. For various degrees of fixedness, we want to optimize I, C, and X. And then we want to find this probability distribution PC given G. And that can be done when you write down this optimization goal, that can be done analytically if you fix certain, I mean, I don't know how technically you want to get, but if you fix, I guess, the cardinality of C, then, you know, you have an expression, an analytic expression based on this optimization goal that you just need to solve self-consistently. Is that?
Thanks.
Okay.
Yeah, I think, the idea, I guess, here is that, so X is your low dimensional embedding, and then the transformation from G to X is maybe very noisy, very nonlinear, but it's not like sort of like a very highly.
No. Okay, good question. Okay, thanks. Yeah, so the thing between G and X is the thing that you have. So that's the thing that you need to put into your system. So the transformation between G and X is the thing that you have. And so it can be noisy if it's what you measure.
Mm-hmm.
And so then based on the thing that you measure between G and X, you now try to infer the best possible mapping between C and X.
Mm-hmm. I see. Cool. Thanks.
But yeah, good. Okay, thanks. I think it'll become maybe a little bit clearer on the next slide, but yeah. Mm-hmm. Thanks. Okay, so with that, I quickly want to introduce the biological data. So I don't know, many of you might have seen this before. This is the fly embryo after about 3 hours of development. What you see here are these striped patterns. They correspond to the segmentation genes that are responsible for developing the fly segmentation along the embryonal axis. So what you see here is that essentially all of these regions between the stripes later on form a different body part of the fly. And so it's important that these stripes are expressed precisely, because essentially almost every nucleus along the embryonal axis is later on going to end up in either a different segment or is going to take a different role in the segment that it later on ends up in. And in fact, the position of what's called the cephalic furrow, so where the head starts, is actually the position of the first stripe. So these segmentation patterns are sort of regulated precisely because they're important, and they're regulated by these maternal gradients. These are signaling molecules that the mother deposits at the head of the embryo and also at the tail. They then diffuse into the fly. They lead to this maternal gradient here, what you see is bicoid. This maternal gradient in 2 or so hours of development leads to the expression of the gap genes. And then the gap genes are also these transcription factors that sort of then determine 5 to 10 minutes later the expression of these parallel genes or the segmentation genes. And so basically on the level of the parallel segmentation genes, we now know that what we care about is the fact that these nuclei take different cell fates so that they are as distinguishable as possible. And the work that my mentors have done in the last couple of almost a decade, probably a decade, is to show that you don't need to know the maternal inputs, but the gap genes sort of provide enough precision for the parallel genes to develop as precisely as they do develop. So the gap genes actually give you enough information for the cells along the axis to take different cell fates. And so with that, we actually have, a system where this framework of the bottleneck immediately fits. So what you see here is again a plot of these measured by Mariella Petkova of these measured gap transcription factor profiles along the embryonal axis. So X 0.1 is where the head is of this fly embryo, and 1 is sort of where the tail is. There are 4 of these gap transcription factors. These are the profiles. They are the inputs. They get measured by the sensors for the pair rule, for the pair rule genes. And then the thing that we care about is the fact that the nuclei can take different cell fate decisions along the embryonal axis. So now all we need to do is apply our information bottleneck optimization scheme and find this probability distribution PC given G, and then hopefully we can compare that to what we think, you know, a sensor in the fly embryo would look like, and then hopefully we can infer something about the enhancer architecture. That's what we've tried. Actually, and maybe, yeah, okay, so one more thing and then I'll take another break for questions. So what one then could do is basically to calculate this probability distribution PC given G. I've shown one of it here, and you know, and I said before that this is what we should compare to the probability distributions that we would expect from other systems. But practically, we don't compare the probability distribution, but we compare its trace on the information plane. And so that's the thing that I want to introduce on this slide. But maybe I should take another quick break for questions in case there are any, and otherwise I'll just go on.
Yeah, I had a question. I guess it's like, how sure are you that you like identified all the gap genes and that like you kind of know all the inputs? And you know, are there other inputs besides just the, you know, concentration of these 4 genes, like or the curvature of the embryo, or it's like other like physical stuff that might matter here?
That's super interesting. Yeah, thanks for the question. So there is a 5th gap gene. I think that's sort of the only other gap gene that is around. But I think in terms of your general question is like, you know, is this really a complete set of inputs, which is sort of, you know, and I think I would say to first order. I would say to first order. It's fine. I mean, of course, you know, there like actually the pair rule enhancers do read bicoid. So there is, you know, that question that, you know, how does bicoid still regulate the, you know, on that level? And I think there are some groups that are looking at that, but to my knowledge, I think I would say it's unclear enough. And I think you can really, like one thing I think that this paper by Mariella Petkova that came out in Cell 2019 in collaboration with all the Princeton folks, I think what they showed there is that you can look at the gap genes in a series of mutants and you can predict the pair genes precisely based on the gap expression pattern. So I would say to first order it works. And then your second question was sort of, are there other like things that matter, like the curvature of the embryo? I'm not so sure about the curvature. I know the temperature matters in the sense that it changes the speed of development, but I don't think it changes the, you know, the patterns. But what's interesting, and I think that's also been a recent thing in gene regulation, is sort of the accessibility of the chromatin. I think that would matter. And these transcription factors, I think, are often uniformly expressed, but they sort of, you know, determine which enhancers, I guess, are open for business. And I think that does matter, and I think it does make it more complicated, but I haven't really, you know, I don't really know of anything of how to introduce that, I guess, on this level of approximation, because what we're interested in here is sort of the question of how do nuclei along the axis develop their cell fates. And so if you have a uniform gradient, not a gradient, if you have a uniform distribution that determines the accessibility, I'm not sure, you know, I'm not sure that matters for this question. And then also, I think in principle, I would say that these even enhancers are open. So I wouldn't necessarily say that but it's a good point. I think that's actually
Yeah, I mean, there's gonna be like tons of mechanisms, like, you know, nucleosome opening and sliding, and, like, you know, whatever like the actual communication mechanism is between the enhancer and the promoter, like, which is still unknown, and you know, it's gonna add a lot of complexities. Yeah, I guess I was just getting back to like this, like, you know, having all the inputs, and yeah, I mean, I guess if you can use gap gene expression to predict like the downstream stuff, that shows you this, like, you know, sufficient. And I'm sort of broadly familiar with this previous work on that, so super specifically, and like, you know, a lot of the conclusions that are drawn is like a sort of like an optimal, it's like, you know, happening optimally. And I guess the sort of, you know, if you don't know all the inputs, it's like you've shown that the inputs are sort of like sufficient in some sense, but the question of optimality kind of depends on, you know, how many other inputs there are or something like that. And
but I mean, I think if you say, I mean, so I think from my work, I think the fact that there sort of sufficient
Yeah.
is all that I think I need.
Okay.
And I think in this previous stuff, I mean, I do think that the sort of fascinating component was, the fact that they showed that they were. So I think in the first paper, Dupuy and all of that, I think they showed that the information was there. And then I think in Marielle's paper, you can think of it as saying that the information is sufficient. And I guess my paper is now like, how can this sufficient information, is it still sufficient after it's being measured? And what can you learn about the measurement by assuming that it needs to be sufficient?
Gotcha.
I think that's sort of but I mean, I see your point about the optimality, and I think maybe we can also get to that a little bit later in the discussion, because it's interesting. I think you're,
Great. Thanks.
So I have a question that's related to the earlier discussion. By the way, I'm really enjoying this talk. It's very clear and really cool area. I wanted to talk about or get a better intuition for in the information bottleneck of why it's subtracting out the mutual information between C and G rather than just using the entropy of C.
I see.
Because I would say that, especially like, for example, when you label here as sensor capacity, you don't want to just have something that might have a lot of information, but it's completely irrelevant to process of interest, and you do want to penalize that. And I'm wondering if there's a particular reason for this choice of having the Lagrange multiplier on the mutual information rather than the entropy of C.
That's a really interesting question. So I think later on I'll talk about not in detail actually, and so it's good that you raise it here. So I mean, we've tried. So I think there are other information bottleneck formalisms that have different constraints. I think here what's important is, so because intuitively, I think when you think of a sensor in terms of these binding sites, then I think what you want to intuitively have a value for is this noise. And so when you think of it in terms of the noise, then I think it makes sense, to constrain the probability distribution between PC given G in some way. So in that case, the mutual information between C and G is actually the right variable, because you're saying I'm measuring with noise. The more noise I measure with, the lower this mutual information gets. And why not the end? So I think biologically, I think in terms of the fact that we think about binding, I think constraining the mutual information is, for me, seems more plausible. And like, I think the thing about the entropy is that actually in a lot of systems, it actually is quite similar, because as you might know, or probably know given the question, the mutual information is always limited by the entropy. And so typically what you find is that the endpoint of these bottleneck curves give you, for the smooth bottleneck curve, in this case, the endpoint is actually the thing that you would get if you compress the entropy, or if you had the constraint on the entropy.
Okay.
So,
yeah, and in this case, you're not worried about like choosing some C that has a whole bunch of other information that's a really complicated thing that's not relevant for this thing, because that's not the regime of like a restriction that is important for this problem. It's really the noise correlation that
right. Yes,
okay.
That's what.
Thank
yes.
you.
Yeah, absolutely. Yeah, thanks for the question. Great, that's great. Okay, so I think if there are no more questions, I'll go on.
There's one more in the chat.
Okay, yes, please.
Why don't I read it? This is from David Falvo. I'm sorry if I missed this, but are you inferring the concentration of the transcription factors using the mRNA expression for these transcription factors, or are you directly measuring their protein concentration? Basically, is G a direct measure of protein concentration, or is it an indirect measure using mRNA expression?
No, that's great. That's a great question. It's the protein. So Mariella measured the, with immunostaining, she measured the transcription factor protein. So I think there's no indirect on this level. It's not indirect. But yeah, thanks for the question. I should have clarified in the experiments. Yeah, thanks. Okay.
I have a sort of sanity check question.
Yes.
Maybe I should ask before I lose completely track. So when we talk about sensor, should I think about like a function with parameters, or with inputs, or is there, I don't know, more abstract than that?
I would literally think of like a measurement device. Like think of a measurement device that counts molecules, like a binding site array, where you say, okay, this binding site array wants to estimate what the external concentration is. And so in terms of the, I guess you wanted to know in terms of like, should you think of a function? So I mean, yeah, I think, so you could think of it as like a counting device. So then the function would be, every count you basically estimate the probability of counting 2 molecules given that there are X molecules in a certain larger volume. And so in that sense, it will give you, some sort of functional dependence on the input transcription factor concentration. Whereas if you think of it in terms of the binding, then with the Hill function, you get something that has more of a threshold type form. So I think both. So I think mechanistically, it's literally like a measurement device. And in terms of
when you're finding the optimal sensor, I guess you don't have to consider, I don't know, like every possible type of, how do you sort of limit your search space?
So, actually, maybe we'll get to that, because I think I don't limit it very much. So I limit it by the data. So when you look at the transcription factor profiles that are expressed here, you can already tell that if you want to measure these profiles precisely, but if the only thing that you care about is for these nuclei to take distinct sulfates, then if you measure, if you measure really precisely where the protein is expressed highly, say at like 0.9, that's a bad idea, because there's like a lot of noise, and also like in each of these genes, and also they basically, the region that they span isn't a really good idea. So that's the idea of a bad sensor, I think, where and I don't need to limit my space, so that because I'm applying the compression onto these profiles, that's basically happens automatically. So the compression basically automatically tries to put where the sensor should measure best. Yeah, I think that's maybe. Yeah.
Okay, cool. Thank you.
Okay, great. Okay, so then I'll go on here. So I think where we were was that I've just said the probability distribution PC given G shouldn't be compared with the sensors individually, but we will look at the traces on the information plane. So what you see here on the x-axis is the information between C and G. So that's what one of these questions was about. That's what I will refer to as the sensor capacity, and it tells you how much does the sensor know about the gap gene transcription factor profiles. On the y-axis, you have the information between C and X. So that's the information that the sensor has about the sulfates. So how much does the sensor have about the sulfate? And then what's important is that not all parts of this information plane are equally accessible. So basically, because of the data processing inequality, a part of this information plane is always crossed out. Very naively, you can think of it as basically, if you measure the information that the gap genes provide about X, then your sensor can never have more information, which is why there's essentially a top bound, and then there's another bound too that we don't need to talk about. But what I do want to do just for the intuition is basically to say that the best possible sensor is basically one that gets all of the information about X, but measures at the highest possible noise level, essentially at the highest possible noise level, whereas a bad sensor is one that measures very precisely and gives you no information about X, just so that you know where in this plane things should be. And so when we now go to all of the gap genes and we apply this information bottleneck approach, we get a curve that's shown here, and you can see that it's actually fairly close to these optimal bounds, so fairly close to the corner. So that in principle, I think is good, because it means that there exists an optimal sensor for all of the gap genes, but it also essentially just says that we were right, I think, maybe coming back to this first question, that the gap transcription factors really give you all of the information about X, so that they are a good enough input set. Because, for example, if you apply bottleneck compressions to neuro data, where you maybe don't have all of the inputs available, then these curves actually often look very different. So they look much lower than the data processing inequality. So that's the first point. But what we really now want to know is, okay, what does this correspond to in the biological system? So what we learn here is that we need a 4+ capacity bit sensor in order to get all of the information, or like, I guess 90% of the information that's provided in the gap genes out. And so in order to give you intuition about what a 4+ capacity bit sensor is, I'll take a step back and I'll talk you through a single gap gene, quickly tell you what we've learned for a single gap gene, and then I'll go back to this for a gap gene network. So I guess I can briefly work again, but probably I can go on just if there's any urgent question. If not, then I'll quickly go on. So let's look at a single gap gene. Let's look at the expression profile for hunchback along the embryonal axis that's plotted here. The information plane is again in the middle. Now you can see that it's lower. So the data processing inequality is lower because hunchback provides less information than all of the 4 gap genes about X. And what we can do now even more intuitively, because it's hunchback, so it's very easy, is we can compare two thresholded senses. So if you think about these thresholded senses, you basically put in a threshold in this concentration space, one threshold or maybe two thresholds. So then you have this probability distribution that has two thresholds, so 3 levels. And you can basically now increase the number of levels, and you will see that as the more and more thresholds you have, the better of a measurement for hunchback you get. So that's what's shown in the middle. Now, what you can do is that you transform this back to the information plane. So all I've done is I've changed the x-axis. The thresholds are still on the information plane. And now we do the optimal bottleneck compression, and we find this line. And so what you find is that this optimal sensor actually exactly corresponds to thresholded senses. And so I should say that when we started this and when we saw this, I think we were all a little bit unhappy. I think Eric hates thresholded senses. Bill, of course, has this interest in Berg-Facel, and I also didn't really like these thresholded senses. And so we were like, why do we do something that's so hard? And then all we get is sort of these thresholded senses. But then when we thought about it, we actually, I think, concluded that it was actually really interesting, because I think what this means is that thresholded senses would be good if they were biologically implementable. But we haven't put anything in that says anything about biological implementation. And so I think what this really shows is that this intuition that biologists had in the last decades about, the French flag model and these thresholds is actually information theoretical intuition. And I think that's important, because when you think about making some of these ideas more precise or more mathematically precise, it's kind of important to make sure that you have the right framework. And so to see that the intuition that's been around for a long time is actually consistent with information theoretical intuition, I think that's a really nice lesson. Okay, and I've seen there's a question. Yes, please.
Yeah, sorry, I was just trying to understand the scales on this plot. So what's the meaning of 2 bits on the x and the y axis where when this thing turns over?
Yeah, so that's because, when you actually look at sort of what's the information that hunchback provides about x, that's exactly 2 bits. So when you kind of, estimate, 1 bit, I don't like these analogies, but 1 bit I think you can essentially think of as a switch. So if hunchback was expressed like that, then, with the switch in the exact middle of the x space, then, you would get a profile that provides 1 bit. In this case, because hunchback, because as you can see, it's, it's better than, I guess, just a threshold. So it provides more information than just a bit. And so that's just these 2.1 bits that hunchback provides. So that's the absolute top. And then I think otherwise I would say, I think the numbers don't really, because it's information. So I think the numbers only mean something, or at least for me, they only mean something if you compare them to the absolute entropy in the variable. So, I wouldn't necessarily worry so much. So you have these bounds, and I think that's what you need to compare to. But otherwise, I think I wouldn't worry so much. So when you basically say on the x-axis, what does the 2-bit means? It basically, again, you can sort of try to say how many switches that corresponds to, and some people like that. And I'm not really sure, because I think it's like sort of simplifying. There are many probability distributions that have information 2 bits that don't necessarily have to correspond to switches, but there is a bound on the x-axis, which would be how much entropy is in your G space. So how many, depending on how many molecules you have, that would be the maximum bound. Does that answer the question?
Yeah, I guess so. Because your distribution of hunchback is continuous, so how much information you get, there's a cutoff there that sort of determines that, right?
So how much? Yeah, yes, I think.
So when you talk about the information contained in this hunchback profile, yes. Sorry, that's probably something very basic that I don't understand.
No, I think
but I just thought, if I compute the entropy, I will have to, in order to do that, I will have to set a bin size essentially.
Yes. Okay, that's right. Right.
And so then I could get any number of bits depending on how wide that is, right?
No, only when you do the entropy.
Right.
So if you try to do the entropy,
but this wasn't the entropy. This was the mutual information.
Yeah.
Sorry. Okay. That
yeah. No, that's a great question. Thanks. Yeah. Okay. So yeah, so okay, so yeah, I guess just to recap, I think I said that these thresholds were information theoretically optimal. And then we did some other things. So then we basically looked at individual sensors. And I think there was a question before, what happens when you compress the entropy? I think this is a system that's similar, but basically what we did here is we basically put in one threshold, put in the second threshold without changing the first, put in the third one without changing the first two. And then we get the sensor evolution that here you can see with these pink little crosses. And that means that, you know, the set of sensors could have evolved hierarchically because you don't need to change the first thing when you throw in the second one. And then the final two things that I want to say about Hunchback essentially are to do with fine tuning of these thresholds. So basically, what you see here is that for a given number of thresholds, when we threw on the thresholds completely randomly, we found that 10% of the sensors with, you know, these thresholds thrown in randomly were actually close to the optimal threshold. And one way I think to see that is, or I guess that just means that not all of the thresholds need to be optimized precisely. And one way to look at that better, I think, is to connect with this idea from sloppy models that Jim Sethna and others have put forward, where the idea is that you look at the optimal and you look at the optimum and you look at the space in the landscape of your parameters around the optimum. So essentially you look at what's the curvature around your optimum. You can do that in this case by diagonalizing the Hessian. So I guess with the curvature of the mutual information between C and X around this optimum. And then you find that the eigenvalues are sort of spaced over several decades. What this means is that there's typically, when you look at this optimum, that there's typically one eigenvalue that basically along which one eigendirection along which everything falls up shortly. So that's something that needs to be optimized precisely because otherwise you lose information fast. But then there are these like sort of eigendirections that are sloppy where, you know, you can vary, but you don't lose that much information or you don't go away that much from the optimum. And that's important because it sort of indicates that fine-tuning isn't necessarily required. And then I guess now this is sort of the point before we go back to all of the genes together. So if we want to now look at this plot for Hunchback, we find that we need a 2+ capacity bit sensor or 5 to 7 thresholds. And so now you can think of maybe an enhancer as, you know, one threshold, so a series of cooperative binding sites. So in that case, you would already need more than one sensor for Hunchback. You can also think of, but like sort of the other thing is that you can think maybe a series of strong binding sites together and a series of weak binding sites make up an enhancer. And so in that case, maybe you can fit the equivalent of multiple threshold-like noisy things into one enhancer. And so in order to estimate that, you kind of go back and think about this occupation model that I introduced at the beginning, where we then put in, you know, the Burke-Purcell estimate for noise. And in this case, because it was just a simple comparison, we try to vary like a lot what reasonable parameters were. So basically the idea is if you have 5 minutes, you know, and a certain binding site array of a certain, you know, 500, you know, binding sites or so, with what noise does this array measure the molecule concentration? You use the Burke-Purcell limit, and with that, I got to about 1 to 3 bit. And this was sort of really the limit. So I guess you can see that if you now take the lower limit and basically say, well, that sensor can only measure 1 bit, you need multiple sensors for Hunchback. But of course, if you're at the higher end and you say a single sensor can give you 3 bit, then, you know, Hunchback is still fine. I do doubt, and maybe that's sort of what's a little bit clearer on this slide. I do doubt that it's on the 3 bits limit because most of these estimates from the occupation sensor ended up in the lower region. But what you can see here is that there are these 2 black stars that are sort of in very different concentration regimes. And these were sort of 2 estimates where I really try to, you know, estimate what is the typical diffusion constant and so on, and where do the sensors then lie on the information plane. And I think what this shows is that really these models, you know, Burke-Purcell and everything, given the measurement precision that we have, it's very hard, I think, to say where exactly you end up. And so I think for this occupation model, the biological noise statistics are really crucial. And even though my estimate is that they are more in the 1 to 2 bit, like a precise enhancer is more in the 1 to 2 bit regime, I think it's still a little unclear. But anyway, so I guess for the rest of the talk, I want to go back to the set of all genes. So let me just recap what we learned from Hunchback very quickly. So we learned that this thresholded senses would be good, so that the information theoretical approach is actually seems to be consistent with biological intuition. We learned that threshold positions could be chosen hierarchically in terms of an evolution perspective. We learned that the threshold positions don't need to be fine-tuned, and we learned that it's likely that more than one sensor or enhancer is necessary in order to measure Hunchback. And so now let me go back to the optimal sensor. Maybe I can break one more quick time for questions and then finish until the end of the talk. Okay, if there are no questions, then I'll go ahead. So now we had this plot before for what does the optimal sensor look like.
I think we did have a question actually.
Oh, sorry. Okay, what's the question?
Sorry, I was just trying to, one more thing. So when you say you pick when you do this thing where you have sort of a random selection of cutoffs, then that's literally just choosing kind of random horizontal lines to cut up your profile.
Yes.
Instead of having the full profile, you just have the gene expression at those random levels, right? Just trying to make sure I understood the procedure. And so you just kind of uniformly sample that. And that's where this thing comes about. Okay, great. Thanks.
Yes. Yeah, that's right. I think the other one is a little bit stronger mathematically, because there I think the point that you made before with the bin sizes, at some point it makes sense that, that would maybe eventually matter. I think here in this regime, we're definitely still fine. But so that's why I like the eigenvalue spectrum. But yeah, thanks for the question. Sorry, I didn't see it. I think I didn't see where that was in the chat, but great, thank you. Okay, so I guess now we wanted to know, what does this 4+ capacity bit sensor mean before? And so again, we can do this comparison with the thresholds. So we can basically compress the optimal sensor into a series of different thresholds that are now actually not one threshold for Hunchback and one threshold for Kupel, but sort of a multi-dimensional threshold compression that basically, I would say, involves different concentration levels for different gap genes. But even in that optimal compression, you need around 50 or so thresholds. So I think that would definitely indicate that you need more than one sensors, because I think that would be pushing it. But then also this 4+ bit capacity for all of the gap genes together, I think indicates that at this point, you really can't do it with a single enhancer. So you do need more than one sensor. And then the other thing that we can do with these multiple sensors is that we can sort of ask, what would it be like if we split the sensor, and if we split the sensor into 4 different sensors, where each of them can only measure one of the gap transcription factors. So assume you have one that only measures Hunchback, one that only measures Kupel, and so on. And then if we now again do this optimization procedure, we find that with that constraint, we get this blue line that you see on the graph here at the lower left, at the lower right, that's always lower than the black line. And so I think what this very clearly shows is that if you split according to gap gene boundaries, you end up always losing information. And so that's definitely not the optimal framework, even though you don't know where on the x-axis your realistic sensor is. So I actually really like this result. And I mean, of course, maybe for the biologists, it's a little bit intuitive, because all this says is essentially that the gap genes also have informations about each other, these expression profiles. And so as a clever sensor will sense in a way that, where that is sort of like accounted for. But I think for me, it was very nice, because also, when you really look at the, I guess the enhancers for these parallel genes, you find that they have multiple binding sites for all of the gap genes, additionally also for bicoid. But I like that, because I mean, often I think you hear this phrase that like, oh, there's so much complexity in biology, in the sense that we cannot, even dream to vaguely understand what's going on. But here, I mean, this shows that this, at least here, this amount of complexity is actually consistent with an idea that would correspond to some information processing optimality. And so I think for me that result was really nice. I know I'm getting sort of close to the end, but I maybe have like 2 more slides on a multiple sensor split, and then does that work?
Yeah, that's great. Go ahead.
Okay. Cool, perfect. So yeah, the very final thing that we're currently doing is essentially to think about what happens if we try to infer multiple sensors abstractly, because the other one we kind of we checked for one particular sensor constraint. Now what happens, or can we infer these multiple sensors together with the information bottleneck approach. And so what you then get is, as opposed to, the plots that we've shown before, where we had just ICG on the x-axis, you now get sort of IC1 for the first sensor and G on the x-axis, and then the mutual information between the second sensor and G on the y-axis. And then the thing that was formerly the x-axis turns into a color scale, essentially. So that's the mutual information that both sensors carry about X. And the first thing that you can see is that actually now the system is a lot more degenerate. So there are many more multiple optimal combinations, of course, subject to constraints, because this was also a specific optimization. But I think the idea is that as soon as you split your sensor, there are many more ways possible. I think, again, this may be like a little bit abstract and maybe a little bit vague, but also I think nice to see so precisely. And then one point that I want to make on the next slide is that for low noise sensors, non-redundant sensors are optimal. So what this essentially means is that in principle, when you have a signal, you could just imagine that you want to measure the same signal again with the same sensor, then your precision goes down by like a square root of 2. But practically what happens here is that, or maybe I should have not switched the slide, it's fun, but practically what happens here is that you can actually use, for these systems, it's actually better to use different sensors if the noise is sufficiently low. And I want to explain this very quickly on a sensor that is a thresholded sensor. So, I mean, maybe, in this case for like 3 thresholds. So if you wanted to split this sensor into multiple sensors, you have essentially first the option to split it into 3 sensors that each have 1 threshold. But so then when you actually calculate the mutual information, you find that this is sort of higher in terms of mutual information between all of these sensors and G than the previous one. And you can see that because if you, for example, say your G is like, say, at 0.9, then you take the result from the first sensor, you take the result from the second sensor, and at this point, you can completely predict what the result of the third sensor is. That's obviously not what you want, ideally, because now your sensors are measuring something redundant. But so what you can do is that you can split it into 2 sensors where one of them has like a sort of non-monotonic activation profile. So this would mean that the sensor would have to be in like, say, off state also for high concentrations of that gene. But then together, these sensors give you, you need both sensors at all times in order to infer the right concentration. And now what's kind of interesting is basically this idea that this non-monotonicity, which I think is also corresponds to these ideas of multiplexing, that can be taken care of in the gap gene network by having a network of genes, because we actually know that some of these transcription factors or genes act as repressors sometimes and sometimes as activators. This happens, for example, for I think NURBS and Hunchback, and of course they. When people say things like, oh, they repress at high concentrations or something like that, then it's of course frequently because also another gene has changed concentrations, and so some of the binding sites are blocked. But I think these ideas again, correspond to this information, something that you would, I guess, learn also from an information theoretical approach. And I think that's it. So with that, I guess I'd like to conclude. So we've looked at this inference-based complement to basically looking at models. We've used the gap gene network as an input. In principle, it works whenever you have an idea for a complete set of, or almost complete set of input factors. We wanted to maintain the output that the nuclei along the embryonal axis need to make the cell-fate decisions. And based on that, we found that the optimal sensors has certain properties that were consistent with biological realities. Also that, this information theoretical approach does capture some of the biological intuition because threshold sensors are good, and that the sensors likely need to be split and not along gap gene boundaries. And so I guess what we're currently working on is a little bit more to push towards connections to models and limits. That's also, with respect to these transcription factors concentrations, can we start again from the models and feed that back into our system? But then also, I guess I've just moved to Delft. So I mean, I should first of all, of course, say that I have these postdoc and PhD positions available. Also, Delft is between The Hague and Rotterdam. So I stole this nice plot from Kees Dekker because I never had like a very clear picture of where Delft was before. But what I'm interested in sort of in general, I guess, in terms of these general questions about precision in biology, is that I also want to keep looking at gene regulation, but then also look at sort of viruses and synthetic systems, because that's something that Delft has a strong focus on. So they have these, synthetic enhancers and, synthetic gene expression. And so I thought, maybe if we learn something from the biological system, is there any chance that we can, sort of apply these lessons to synthetic gene expression? and so, yeah, that's, I guess, it. So yeah, I should thank you for your attention, and I should, I wanted to acknowledge again the collaborators and then the Princeton teams especially, which, yeah, it was a good time at Princeton. So yeah, thanks very much for your attention. And,
thank you so much. Sorry, that was very good. That was a lot of fun. For any questions on the talk, people can feel free to ask them now. I was kind of wondering, like, what would you do in the situation where you don't have that gap gene network? Like, can you use this sort of framework to infer new networks from, like, you know, omics data sets, or would that be a little bit infeasible right now? I don't know.
So I think you need these two ingredients. You would need to know what you think these genes provide information about. Ideally, actually, and I think this would make the system here simpler too, is if you had a set of output genes. So if you had, and the reason why they use, or why we've used X here all the time, is basically because the parallel genes, you can't measure all of them together in the same embryo. And so the output is kind of unclear. So in a way, I think, or like it's clear in this system because we have the biological intuition. So in a way, these omics data, I think, would actually be really fascinating. But then I think you do need something that's to first order complete, because I think I was going to say it doesn't really make sense when it's not. But then on the other hand, I think that like a lot of these neuro data sets, I'm also never really sure whether they are really complete. So I guess you could, I mean,
nothing's ever complete.
Yeah. Yeah. But yeah, I think that would sort of be one thing. And then I think I do in principle think that maybe information theoretically you can do maybe a little bit more in these omics data set. But yeah, it's tough, because I think if the data is also not precise, then so if the data doesn't have mean and noise, then it's that makes it difficult.
Yeah, I hear that. Janne?
Yeah, that was really wonderful. Thank you for the talk. I was just curious. So yeah, I was actually thinking about synthetic systems, and so I guess have you thought about that? Like, what would be if you could just kind of wave a magic wand and create the ideal synthetic system on which to try out these ideas? What would that be? Like, how?
Yeah. Okay. So I mean, there are actually, I think, several. Like, I mean, okay, so I actually just recently talked to someone who was a little bit like non-encouraging about the synthetic idea, even though I still think that it should work, because I think that the problem often in synthetic gene expression is that it's frequently that the system overall is worse than if you just put something into a bacterium. And my understanding is that no one really has an understanding for why that's the case. So I think
sorry, when you say it's worse, you mean you reduce fitness of the cell?
Yeah. Yeah.
Well, yeah. So we've actually figured out ways of avoiding that with
really
with Mo. Yeah. So actually, we meaning not me, absolutely, but Mo Khalil at Boston University is going to soon come out with a paper where he will show how to control fitness effects, at least in yeast, when you introduce certain synthetic systems. So yeah, so no, I mean, don't listen to people. I mean, people. No, just there's like a million ways that things can go wrong, but that doesn't mean you should try it. So anyway, so let's assume that you can control fitness and that you won't lose your synthetic system as the cell divides, if that's the issue, because that I think we're getting to the point where we understand, at least in some very simple systems, how that might work. Yeah.
Oh, that's super interesting. Yeah. So I mean, I think basically I would say I would have sort of two questions. So the first is, like, I think what I would like basically, can one, predict something about enhancer architectures for gene expression in these synthetic systems? So that would be one thing that I would be really interested in the sense of like.
Wait, now I'm really confused. I mean, you're building a synthetic system so you can kind of create the architecture you want. I mean, that's
yes,
so I'm not sure what you mean by predicting.
Well, in the sense that, like, my understanding is that often in a synthetic system, so when you then put the codon sequence in, or you put the promoter, or you pick what are the binding sites. So my impression is that experimentally you sort of try through, or you maybe try, you look at E. coli or like the gene that you want to express, and you copy from them. But so basically, can one sort of go in and say, maybe based on the codon sequences in the organism that you're trying to express in, or in like a cell-free system, what would give you the highest yield in terms of the codon sequence of the enhancer? Or, I mean, I wouldn't even dream, but maybe in the long run, even the spacing of the enhancer away from the promoter. Like, what type of sequences give you the best yield for the gene expression? I think that would be sort of my dream, I think. And then, of course, like in synthetic systems, the way experimentally, I guess one would.
Sorry, when you say best yield, you want to connect the yield to some computation that this enhancer does on the chemicals that it's exposed to. That's what you mean by yield,
right? Or I would say, maybe there is some sort of signal, and then I want the cell to express a particular protein as in some sort of functional form, depending on the signal. What's the best enhancer that achieves that?
Right. So you can compute that, right?
I can't compute that yet. And I think I would really like data sets. I mean, I would really like to figure something out, like learn something about data sets that would help me to, maybe make this methods or other methods more precise.
So the ideal experiment might be, for example, to move locations of binding sites around, or to change the affinity of the binding sites for the transcription factors, and then for each of those constructs, see how the input-output changes, and then do something like that. Okay.
Yeah. Mm-hmm.
Yeah.
Or
now,
shift,
whether you're in the position where you can make a prediction for an experiment, so like that they could actually go in ahead and test. I understand how doing those kinds of experiments would be a cool, sort of a cool playground for these ideas. But I was just curious whether this is at a stage where you can say, well, if you make for me this kind of an enhancer with these kinds of elements, then I expect you will get a really good, readout, versus if you do it another way, you won't. Like, am I in a position to do something like that?
That would be my dream. I don't think that I could do it yet, because I think for now I would probably try to use some data that would be available. I would try to infer, apply this optimal sensing approach and say, given that, what is the best, what type of structure does it have? But then to kind of predict, because my stuff doesn't even, use at the moment any of the codon sequences. But that's, I think, sort of the direction that I would really like to move towards. I think that I think would be really exciting. Yeah. Mm-hmm. Yeah.
Okay. Thanks.
Yeah.
Fun stuff.
Yeah, the other thing where the synthetic systems might help is because I think you could sort of check maybe more locally what like droplets around enhancers, whether there are any, what they look like, how they respond to different concentrations. And so that might also give you a better estimate for the noise around a particular enhancer. So that's also where I think these synthetic systems might be cool, because you sort of can just tune it, whereas in the fly, I mean, it's a little bit tough to measure. Yeah. Mm-hmm.
So I had a question and also a comment on this previous discussion. So I guess I'll start with the comment first. This idea of trying to, like, if you like just concoct some kind of like regulatory architecture and trying to sort of predict what the input-output relationship would be between like location of the enhancers and the strength of the binding sites and all that kind of stuff, and then like what does gene output look like? That's kind of like one of these like holy grail type questions. And I would say, like, if you can do that, then that means that you've like really understand something going on. You understand like something about enhancer mechanisms, or you understand like something about, like, you understand it at like some level of representation that is like very useful. And I mean, I think we're close to like being able to like you can write these models down. You can like write down like one of these like Markov process type models of like a gene is regulated by one enhancer or two enhancers, and say like, oh, okay, like these on and off rates are like dependent on like the, say, like chromatin accessibility or something like that. And
sorry. No, I wanted to say, because to my knowledge, I think it's still super complicated. So I have one colleague here, Christophe Danelon, who basically looks at gene expression in a cell-free system. So he uses this E. coli extract and he puts like a plasmid in, I think, and then this like they are looking for the protein. And I think all sorts of stuff goes wrong that I hadn't even dreamt of. Like, I think the ribosome doesn't load, like stops, because it's not sort of optimized in quotation marks to that context. And then I think even in the fly embryo, which of course is much more well studied, I think there are these, another collaborator of mine looks at the wing patterns, and this is Nicola Gompel in Munich, and they basically find that, there's also an enhancer region that creates this wing pattern, but basically, you change like a few binding sites and you have essentially no idea what the output pattern is like. So you change it here, and then, it's just, because I think also maybe in relation to your previous question, because it depends on, whether there's another transcription factor, maybe even a uniformly expressed one that happens to bind there, that happens to then block this binding site. So I think, we are unfortunately still a little bit far. I mean, at least,
Yeah. I mean, as soon as you start, adding the complexity just increases like so much. But even like, I mean, you're in the Netherlands, right? Like this like paper from Tineke Lenstra, where there's just like one TF in yeast, like California, and doing the imaging and just like seeing the complexity for a single transcription factor, and a single gene output is already quite complicated.
I
guess my point was more just like, can you build a model that does that? And, sure. Like,
right. I see.
I think the prediction, like, I mean, I have that. I'm happy to like chat with you about like that
cool.
So I guess I was just saying, like, from a like straight up, like, can you make a prediction about like what the input output relationship looks like? I think we're there.
Oh, that's super cool, actually. Okay.
I guess my question related to your talk was about this idea of like trying to like learn the regulatory architecture of like how many enhancers are there, and like do they share like common TS just based on the like what you observe about the gap change. Is really interesting. I guess my question is like, what do you know experimentally? Like, the even enhancers, where they are. Like, do you know like what combination of TFs are binding there, and like what is like, can you compare like what is known experimentally against what you're finding here?
So exactly. So the Eve enhancers, but only for Eve, not for all of the other parallel genes. I think Runt is much more complicated, and with other ones, I don't even know. But for the Eve ones, I think Mike Levine and Stephen Swan and Angela DePace, they've also like sort of characterized the binding sites. This was sort of an old paper, but I think it's sort of also not entirely clear whether that captures all of it. But I think, what this shows is that there are multiple binding sites. What I can't do with my method is that I can't say, oh, the binding site for giant should be here, or like the binding site for cupula should be here, because the model, like, it's not, I guess, on the level of a model. And the other thing is that, I guess, because it's all optimized within the fly embryo context, I couldn't really say, it's very hard for me to kind of predict a variation. Like, I couldn't say with this method, oh, like this other binding site should actually be better, because then it's likely that in the fly embryo, it's still not going to be better because it's already sort of good enough. So, because you would basically tune one node of a network when other nodes of the networks are sort of optimized for a particular thing. So if you just tune one thing, then overall the result, I think, would still be worse. So I think it's a little bit hard to kind of find a precise test, I think, in the fly embryo context. And I think that's why these synthetic systems might be cool.
Okay, why don't